Good evening, T.Y.B.Sc. student. Today we are going to discuss on paper 4th, BO 334, semester 3rd, spermatophyte and paleobotany. In last lecture, we already discussed about the male cone, female cone, then the structure of microsporangium, structure of megasporangium, megasporophyll, microsporophyll. This everything is discussed in this syllabus. Then today we are going to discuss on the topic that is the sexual reproduction in pinus. Since the plant body is heterosporous, it bears or it have the both microspores as well as the megaspores. Both are present on two different cones, and these cones or these strobilus, they are present on two different branches, which will develops the microsporangium and megasporangium. On its germination, these microspores and megaspores will form male as well as female gametophyte respectively. So at first we are going to concentrate on the male gametophyte. The male gametophyte is started from the winged pollen grain or microspore or the spore simply. Why it is known as microspore? Because it is male, so it is known as microspore, and as it has the wings, so it is known as winged pollen grain. When we observe the structure of this pollen grain, this pollen grain is winged pollen grain. When we observe the pollen grain, this is the pollen grain of the pinus. In last lecture we already discussed about the pollen grain of the pinus. So this is the pollen grain, and these are the two wings. So it have the three layers, that is outer layer, then middle layer, that is exine, mesine and intine is there. So at first what happens at the time of formation of the male gametophyte, the microspores at the maturity of the microsporangium or at the maturity of the male cone, the microsporangium splits open. There is the slit for the opening, and microsporangia gets open, and number of the microspores are discharged, number of microspores are liberated and transported with the help of the wind. Then these microspores or the pollen grains or winged pollen grains then reaches to the small pollen chamber by drying the pollen drops. When they reaches to the pollen chamber, that is towards the gynoecium, or we can say that towards the female part, that is megasporophyll, or towards the ovule. So at that time, when they reaches to the small pollen chamber by drying the pollen drops, then the starting of the male gametophyte takes place from this stage. So each microspore will start to germinate, and first of all they gives the two cell structure. Means this cell divides into the two cell. The one cell is generative cell, that is this cell divides into two cells, and the upper small cell is nothing but the generative cell, and the small that is large cell which is present below, it is nothing but the tube cell. So what happens, that is this tube cell performs the function of the formation of the such type of the tube, which is known as the pollen tube, and this generative cell divides into the formation of the two cells. These two cells are nothing but the one is body cell and the second one is the stalk cell. Means what happens? First of all, the cells are divided into two, like this one. That is, firstly, this single cell, that is each pollen grain, each winged pollen grain is the uninucleated and they are tri-layered. That is, three layers are there. So uninucleated, tri-layer is there and having the presence of the wings. So these tube or these nuclei or this single cell divides into two cells. One is generative cell and the second one is the tube cell. This tube cell is responsible for the formation of the such type of the tube, which is known as the pollen tube. And the second cell, which is present above this cell, which is present at the back side, it is known as the generative cell. So this generative cell again divides into two cells. This generative cell divides again into two cells. Which are those? That is, one is the stalk cell and the second one is the body cell. So tube cell remains as it is, which are growing above or which are growing faster. Then stalk cell remains as it is, but laterally the body cell again divides into the two cell, and this body cell is converted or it divides into the two cells which are nothing but the sperms, or they are known as the male gametes. These male gametes are non-motile in nature. So this is the formation, that is entire structure is known as the male gametophyte. This is the male gametophyte, and this male gametophyte is responsible for the formation of the sexual reproduction at the further stage. Then female gametophyte. This female gametophyte is formed, that is, this one is the ovule, and this ovule is of three layered, and this cell is, or this portion, which is the large portion as compared to the other. So this large portion is nothing but the nucellus, and here the one functional megaspore is there inside the ovule. This is the actual structure of the ovule. This is the that is funicle part. This is micropylarate. This is the chalazoid, and opposite to this chalazoid there is the micropylarate, and through this micropylarate the pollen is enter. Pollen start to germinate, and they have the presence of such type of the two sperms, two gametes. So this is actually male gametophyte. This one is the pollen tube, this one is pollen tube, then two gametes are there, then tube cell is there, and one is the stalk cell is there, and body cell is converted into the formation of the two gametes. So such type of the structure is formed. Then after that, this single megaspore which is present here in the nucellus, which is present at the center, this single megaspore divides and redivides, and again it undergoes the pre-nuclear division. So what happens in this way, continuously when the cells are formed or nuclei are formed in the nucellus, they are divides and redivides by the meiotic division. And as meiotic division takes place, then what happens? That is, number of cells are freely formed, that is about the 32 nuclei are formed in the nucellus, and at the center there are the formation of the such type of the vacuole, and these 32 nuclei gets migrate towards the periphery. So free nuclear division will take place in the next spring very rapidly, and again the number of nuclei goes on increasing up to 2000. So what happens when the 2000 nuclei are formed? the two archigonia are produced at the apex of the female gametophyte. Here, at the apex of female gametophyte, there are the formation of the two archigonia are formed, and this entire structure, that is when there are the this space is occupied by the entire nuclei, this is nothing but the female gametophyte. Then archigonium. This archigonium is formed when you observe this structure, that is this one is the megasporophyll, and this megasporophyll have the such type of the stalk, and this one is the bract scale, and this one is the ovuliferous scale, and to this bract scale and ovuliferous scale there is the presence of such type of the ovule. So these ovules are two on the either side at near to the this bract cell or bract scale. So they are present on either side, and this ovuliferous scale is pointed and inwards at the top. In this way, there are the continuous formation of the such type of the cells are takes place. So these cells are divides and redivides by the meiotic division. Then these nuclei are migrated towards the periphery. Again there are the further meiotic division is takes place, and this cavity is formed, and these nuclei are migrated towards the periphery. In this way, the entire this ovule gets covered by the such type of the number of nuclei. Then the cell which is present at the periphery, which is known as archigonial initial cell. This archigonial initial cell is the peripheral cell, or it is subhypodermal cell or below to the hypodermis. This cell divides and redivides into number of cells. So first of all, this cell is archigonial initial cell. This cell divides into two cells, that is the stalk cell, or we can say that the neck cell, and below to that there is the ventral canal cell, and at the base there is the this venter cell. So this ventral canal cell or this ventral canal cell forms a venter, and this basal cell, which is known as the egg cell, this egg cell is comparatively large, but there is the absence of neck canal cell. So when there is the mature archigonium is formed, this mature archigonium is formed like this one. So at the center there are the spindle fibers are found there, and there are the number of the cells are present here, that is the paranuclei, we can say that these are there, and at the center there is the large nucleus or the egg nucleus which is present and which is covered by the jacket cell. So the ventral canal cell is present here, then the neck canal cell is there. This neck canal cell is not furtherly growing, but at the center there is the egg cell is there. So this egg cell is fertilized when the sperms are enter inside the such type of the spore, megaspore, and then there are the further reproduction is takes place. So at the time of the pollination, the pollen grains are light in weight, and hence the pollinating agents never visit towards such type of the cone, that is the male cone, because the flowers, that is the cones, are not so attractive, and as they are not so attractive, so pollinating agent never reaches towards this one. So what happens, that is pollination is takes place with the help of wind, and hence it is known as the anemophilous type of the pollination. So once the pollination is over, the pollen grain develops the pollen tube, which will penetrate in the nucellus and reaches the neck of the archigonium. This it reaches to the neck of Archivonium, and the tip of pollen tube contains two male gametes, and the tube and the stalk nuclei. That is, the tube cell have the tube nuclei and the stalk nuclei is there, and body cell or the body cell is converted into the formation of the two male gametes. The pollen tube comes in close contact with the Archivonium, and the tip ruptures chemotactically and discharge it contents into the egg, that is inside this egg. That is, when it reaches towards this one, everything takes place inside the such type of the ovule, and ovule is attached to the megasporophyll. Megasporophyll is the leaf-like structure. So everything happens in this one. Then it reaches to the it discharges the content to the egg. One male gamete approaches to the egg nucleus and unites to forms the zygote, which is diploid one. The second male gamete with stalk and tube nuclei degenerates, or they gets vanish, they gets disappear. So fertilization which is completed by such way, it is the single fertilization. As like the angiosperm, it is not a double fertilization, but it is only the single fertilization, because the stalk cell, tube nuclei degenerates. And generally the fertilization in the pinus, which takes place in the end of the month of June, that is when the environmental condition is suitable and when there is the humidity and moisture in the environment. At that period, the such type of the fertilization takes place, and when the fertilization takes place, the end point of this fertilization is the formation of the seed. Then mature embryo is there. That is, what is mature embryo? Mature embryo is the content of the seed. When we observe the embryo, this embryo is the straight in position. When we observe the structure of this embryo, it somewhat looks like as a bell. That is, it have the pointed suspensor, and the entire structure is somewhat looks like as a bell. When we observe the basal part of this one, it have the presence of 8 to 10 slender yellow cotyledons at the end. When you observe this one, it have the 8 to 10 yellow cotyledons, and these cotyledons are responsible for the further growth. Away from these cotyledons are present away from the micropylar end. Means this end is the micropylar end and this one is the chalazal end. A large hypocotyle below the cotyledon and minute conical stem apex, that is the plumule is there. That is, this plumule is present near to the cotyledon, and plumule is responsible for the formation of the shoot in the further stage. So plumule are known as the shoot initial, and radicle is the root initial. Furtherly, radicle are responsible for the formation of the shoot, and the tip of the radicle is attached to the dry coiled thread-like structure, which is known as the suspensor. This radical tip is attached to the suspensor. This is the actual structure of the mature embryo. Then structure of the seed. When we observe this structure, the seed is made up of, when we observe this entire structure, this seed is made up of it have the following or different parts, that is the seed coat, tegmen, then perisperm, endosperm, etc. Then first of all, seed coat. What is the seed coat? When we observe this seed, this is the LS of seed and this is the entire seed. Seed is the result of the fertilization. When the pollen male gametophyte is formed and male gametes unites with the egg, then their result is the formation of the seed. So when the seed is formed, the seed when gets mature, seed becomes hard. At that time, the seed have the different types of the parts. That is, it have the outer seed coat, which is outer, and hard, which is the covering of the seed, known as testa, or in the language of botany, it is known as the seed coat. It is formed only from the middle stony layer of the integument. That is, integument is made up of the three layers. The outer layer and the middle, that is inner layer, are comparatively fleshy. But third layer, which is present at the center, it is the stony layer. And this stony layer is responsible, this stony layer of the integument is responsible for the formation of the seed coat. Then when the seed coat is formed, then outer and the inner fleshy layer gets disorganized, they gets disappear at the stage of the maturity of the seed. The seed coat protects the inner delicate parts of the seed from the drying as well as the mechanical injury. Different types of the pathogenic attack or the drying, it is get protected by the seed coat, because seed coat is hard as well as it is protective in nature. So mechanical against the mechanical injury, against the pathogenic attack, or against the drying, the seed gets protected by the seed coat. So this outer structure is known as the seed coat. Then next layer is tegmen. What is tegmen? It is brown papery inner seed coat next to the testa. Means inside this seed coat or inside this testa, there is the papery covering, which is known as the tegmen. Sometimes these wings which are present in the seed or in the winged pollen grain, these wings are also remains with the seed, or sometimes these seeds are known as the winged seeds. Then third portion of the seed is the perisperm. The nucellar tissue, the perisperm helps in the providing nutrition to developing embryo. What is it? That is, this is the central portion, and this central portion, which is comparatively it occupies the more area, and this perisperm is the nucellar tissue, and this nucellar tissue performs the function of providing the food material to the developing embryo, as like the endosperm which is present in the angiosperm. So as like the angiosperm, there is the perisperm is there, and perisperm provides the food material for the developing embryo at the further stage. Last one, or the last portion, is the endosperm. This in the center of it, there is the distinct central cavity called as corrosion, in which lies an embryo. The newly developed young sporophyte is differentiated into the radical, plumule, and cotyledon. It have this embryo. This is the entire structure of the embryo. It have the cotyledon. It have the plumule. It have the radical. Such type of the structure is present in the embryo, and it have the presence of number of the cotyledons, which are 8 to 14 are there. This is the ELS of the seed, which is the actual structure of the seed, which is found in the ELS. So this is sufficient for today. So thank you very much. Stay safe, stay home, have a good evening, and thank you very much. Okay, thank you.